Morning, everybody. Welcome to Grand Rounds. Next week, our speaker is going to be one of our residents, Chief Resident Angela Coombs, and the title of her presentation is going to be Let's, in parens, Not Talk About Race, the Challenges of Open Exploration and Cross-Racial Therapy. And the discussants will be Aaron Relaford and Dion Powell. I'm going to suspend making announcements today just to give us the full amount of time to spend with a visiting dignitary from Washington, Dr. Susan Koester. And she's been in Washington a long time, and prior to that, she was in St. Louis, where she did her postgraduate training, but she has a lot of connections to Columbia. She went to undergraduate school here. At WashU, she was there when Jerry Fishback, our former dean, was the chair of neurobiology. He was the dean that recruited me. That was his last recruitment before he was asked to step down. I don't think there's any connection. And Roy Vagelos was formerly the chair of biochemistry there, who was, of course, our illustrious chair of the Capital Campaign and donor of all things important. And last great physician, scientist, CEO of a major pharmaceutical company. And also, she's related to, unbeknownst to me, to John Koester, who held forth as the chair of neuroscience and the director of the Center for Neurobiology and Behavior, who resided along with Eric Kandel and a host of other faculty in the Kolb building for many, many years. That sort of tenure has come to an end. They've traded up. But it's really nice to see him back with us. John, you want to stand up, take a bow? He looks a lot younger than he did when he was in that role. Sue comes to us from Washington, and let me just tell you, particularly for the younger people in the audience, you better sit down, this is going to be a little bit long. In terms of your professional academic careers, particularly if you're going to be pursuing research, the two most important people in your life are your mentor and the program officers at the NIMH in the extramural division. And they're, in a way, the unsung heroes of the biomedical research enterprise, because they almost always began as scientists, but by dint of some circumstances found their way into the institute and to the extramural program, where they're really in a position of policy and funding decisions and strategies. And I remember the first grant that I got, remember what I'm talking about? Ellen Stover was the program person, and she did psychotherapy with me on the phone. She didn't bill me, didn't prescribe me, but it was very helpful, and it can make all the difference in the world in terms of helping you really navigate the grant submission and review process, and guiding you in terms of where the scientific opportunities are. And they don't get the credit and the recognition, but that's the highest form of contribution. And also, they are more enduring representatives of the NIH, and the directors come and go, and they have to sort of adjust to the different directors and learn how to manage up. But they're really just a tremendously important and valuable resource. So my advice to everybody that I mentor, teaches, make your program officers your new best friend for life. So at any rate, Sue is currently the Deputy Division Director at the Division of Basic Science Research at the NIMH, and works closely with Linda Brady, who many of you know very well. She began at Columbia, as I mentioned. She did her PhD at WashU. Her thesis was on differentiation of the efferent projection neurons of the mammalian cerebral cortex under Dennis O'Leary. She did a postdoctoral fellowship at the Salk Institute in California, and her research was focused on embryonic development of the spinal cord. You have to mention somewhere along the way if that device that I saw, which was implanted and enabled spinal cord injured patients to walk, is really a tremendous breakthrough or not. But Sue's had a variety of positions in areas of research, and she really has earned her scientific bona fides. She has been a scientist in the academic community. She was in the intramural research community. Her research was on developmental neurobiology. She has been involved with the programs initiated by the Division of Basic Research, including the, is that the DNA repository, the program? Is that the one that's in Rutgers, though? Or in New Jersey? That's a different? Okay, well, the NIH Common Fund Genotype Tissue Expression Project. Her division has an annual budget, which is allocated extramurally to a variety of grant mechanisms of $350 million. And the research ranges from human genetics through molecular, cellular, integrative, and systems neurobiology relevant to mental disorders. So she's really well positioned to give us her perspective on the field of neuroscience as pertained to mental illness, and also how it can be translated most effectively into clinical application and the ways in which the NIMH is seeking to try and support such research. So please join me in welcoming Dr. Susan Koester.
Thanks so much. It's wonderful to be back at Columbia. I know many of you have gotten probably career advice from my Uncle John. I also got career advice from him long ago when he said, as long as you're visiting Princeton, you might as well come across the river and tour Columbia. I hear they're going co-ed, and that worked out pretty well for me. So he gives good advice. I'm going to appreciate the opportunity to talk to you today about NIMH and what we're doing and what we're interested in out here. I want to give you an overview of what I'm going to talk about, a little bit about our strategic priorities, a few new initiatives that we have coming down the pike, a brief update about the BRAIN initiative going outside my institute to broader programs, a couple of common fund programs that I want to mention that I think should be of interest to people in psychiatry. Something about our training mechanisms. I know that's of interest to a number of people here. And then finally, I'll finish up with what I hope is good news with the budget update. So starting out with our strategic priorities. We know that the mission of NIMH is to transform the understanding and treatment of mental illnesses through basic and clinical research, paving the way for prevention, recovery, and cure. And we're working toward that with, through our strategic plan for research, which is divided up into 4 strategic objectives. I know all of you have studied this very well. It's been published on our website for a few years now. Objective 1 is about defining the mechanisms of complex behaviors. That's mostly our basic and early translational science objective. Number 2 is about charting mental illness trajectories. That's thinking about mental illness in terms of neural development and brain development, determining how and when we can intervene. Objective 3 overlaps a little bit. It's about prevention and cures, treatment development. And objective 4, strengthening the public health impact, is where we really think about how to disseminate interventions that work into the broader community. So in September 2017, a year ago, NIMH posted its annual update to our strategic research priorities. You might notice the word annual. We are currently working on our next update. When I go back, I'll get to go to a number of meetings about that. Last year, we provided revised guidance about encouraging computational approaches and emphasizing rigor and reproducibility, as is happening across the NIH. And of course, highlighting data sharing and common data elements that make shared data easier to use and compare across studies. So the strategic research priorities are a place to take a look if you're wondering, broadly speaking, what the NIMH is looking for in terms of new programs. Let's see. So Jeff mentioned I come from the basic neuroscience division, down at the basic end of the translational pipeline. We have 3 major research divisions. The next one is Division of Translational Research. Some of you, I'm sure, know Holly Lisenby, who manages that division. And our Division of Services and Intervention Research is responsible for taking new treatments and new interventions to the clinic for large-scale efficacy trials and helping disseminate those to the public. We have a number of cross-cutting offices. Actually, it's a division on AIDS. Like all institutes, we have a separate AIDS budget. Ours ranges from basic neuroscience of the virus in the brain to understanding behaviors of populations affected by the epidemic. We also have offices that look after our genomics programs, technology development, which plays a big role in both the BRAIN initiative and also if you've looked at the National Database for Autism Research, NDAR, that's where that's housed. And we have an office that's been reorganized, reconceptualized for disparities research and workforce diversity. So that's a little bit about where I come from. I wanted to talk to you about research priorities. These are the priorities that Josh Gordon initiated when he came from here to us. Thank you very much. Most urgently, he's been very eager to boost our efforts in suicide prevention and early intervention, to identify implementable evidence-based practices and as well as knowledge gaps. We also an interest, as many of you know, in neural circuits, trying to figure out how to interrogate neurocircuits and to use that information to ultimately intervene in disorders on a circuit basis, and then bringing computational approaches to improve our understanding of mental disorders and of basic neurobiology. So suicide prevention, I don't have to tell this audience that this is a national health crisis. The age-adjusted suicide rates have been going up. This is both in male and female. We have a number of initiatives that are ongoing, research projects. One that's, oops, whoa, that's a little fast. One that's got another year on it, and then projects that are research projects that are ongoing both in terms of implementing existing interventions in the clinic and also addressing research gaps, mining existing data sets. So in terms of understanding neural circuits, we conceptualize our disorders as disorders of circuit dysfunction, driven by an interaction of genetics and experience that's instantiated through molecular or structural pathology. So we want to understand both brain function and dysfunction, characterizing the molecular identity, anatomy, activity patterns of circuits in a very cell type specific manner. This interacts with things that are going on in the BRAIN initiative that are looking at cell types in undiseased brain, both in model systems as well as in humans. And we are trying to leverage that information to go into psychiatric populations. And then, of course, we also want to be able to monitor and manipulate circuits to improve function. One of the ongoing initiatives that is managed out of my division is to use what we know about physiology to try to improve the predictive value of preclinical assays in therapeutic development. So we have a number of behavioral assays that researchers have used for a long time that have not proven to be predictive in terms of treatment development for our disorders. And so this PAR, which started out as an RFA, is looking for proof of concept studies to identify non-invasive neurophysiological measures that we can show that they are coherent or analogous between humans and non-human species, that we can go into the physiology in the model systems, and then also compare these, use them as an assay potentially in humans and ultimately in clinical populations. We have a concept that's recently been cleared by our council that to take a step back from that stage of assay development to try to develop new neurobiological measures in animals that reflect fundamental brain processes that would contribute to mood or cognition. Measures should be amenable for study in awake behaving animals and then ultimately subsequently in humans. We want to be able to validate those measures, that they are changeable by things that, manipulations that we do to the animal, and that we can evaluate the performance, test-retest reliability, performance in terms of an assay. So one of the examples, this is an initiative that is under development with Lois Winsky in my division. One of the examples that she uses, for example, as a physiological measure are sleep spindles. The spindle generating circuit is well known, well understood, and translates easily between mice and humans, and could potentially be validated as an assay going forward. In terms of computation, we have a new RFA out on the street looking at computationally defined behaviors in psychiatry. This is focused on human behaviors to develop computational approaches to parametrically detailed behavioral assays across mental health relevant domains of function. I asked Michela Ferrante, who's the scientific contact on this initiative, give me an example. What are you talking about, a parametrically defined behavior? He said, we have a few of these that are very well developed, like reinforcement learning or reward prediction error, that have been modeled really well. So that's not what we're looking for here. What we're looking for is new behavioral assays that one could develop and computationally model like that. And so if you are interested in that, if that sparks an idea, please get a hold of us. And for each of these initiatives, I should say, they always have an RFA means that there's money set aside, or a PAR. You can actually put that number into your Google search box. It pops right up really easily. And down at the bottom, there's a scientific contact with a phone number and an email address. Please get in touch with us, because we can explain better what we're looking for. And if you have an idea that you're thinking of applying to one of those, we would love to talk to you about it and help you develop it in the direction so that we're both thinking the same thing. And that's my message throughout the talk is please get in touch with us. I have a whole building full of people who are excited to talk to you. I wanted to talk about a couple of the work groups of our council who have been giving advice about directions in particular areas of our field. The big one that came in last winter was the Genomics Council Work Group. A report that was predominantly about human genomics and our studies on human genomics and new directions. You know that there's been an explosion of findings in genomics of psychiatric disorders, due in large part to the combination and comparison of multiple data sets through the Psychiatric GWAS Consortium, the PGC. And so we asked council to take a look for some time about the next steps and the next directions for where we need to go with all of this information. So most of their recommendations are reasonable, straightforward, and in line with what we would hope to do. Future studies in genomics should use appropriate and rigorous statistical methods. We're very much in favor of unbiased association studies. We have an unfortunate history in psychiatry of picking candidates for study based on face validity and being wrong. So, or learning a lot, but not learning what we need to know to move forward. And so unbiased association studies, looking at all types of genetic variation, both common, rare, genomic, epigenomic, for disease association. Looking beyond DSM diagnoses, really taking an RDoc-focused approach to genomics, as well as we do for our clinical and other translational trials. Looking at genomic and phenotypic variation across diverse populations, both different populations in the US as well as globally. Of course, developing and sharing research resources. Genomic community has been a leader in that, and the success of the PGC is a really great, I think, testimony to the value of data sharing broadly. And then finally, the one that has created some heartburn in the community is using robust genome-wide significance to choose genes for further study in model systems. And so we have been taking these findings and the full report and Josh Gordon's blog about the report are all up on our website. And if you are thinking about doing genomic follow-up, I encourage you to read those and get in touch and talk to us about what you're thinking, so that we can make sure that we're in line, because we're using these recommendations to make funding decisions. We also have a work group that has a clever acronym that I can't for the life of me remember, but it's about changes to the matrix of RDoc. And their charge is to provide a focus for standardizing submissions for suggested revisions and to make recommendations to council when we need to change the RDoc matrix. They met, they had workshops, and they have recommended the addition of a sensory motor domain to address a very important component of a number of clinical symptoms, looking at motor actions, agency and ownership, habit, and innate motor patterns. I need to emphasize that this is a clinical domain, that the clinical divisions are going to be looking for projects along these lines. In basic science, the sensory motor domains are very well covered by other institutes, and so my division is not looking to build a basic science portfolio in motor behavior. So again, please talk to us if you're thinking about a project in the motor domain. So I'm going to shift gears a little bit now and move outside the Institute and talk a minute about the BRAIN Initiative and where we are with that. Everybody knows that BRAIN 2025 was the report that presented a vision for a large initiative that crossed multiple NIH institutes, as well as involving NSF and other agencies around the US government. We are focusing on circuits, networks, measuring fluctuating electrical and chemical patterns within the circuits, and then, of course, to understand how this helps us generate unique thoughts and actions. The BRAIN Initiative is focused on normal brain activity and behavior, and then the individual institutes are hoping to leverage what comes from the BRAIN Initiative to focus on their specific disorders. The latest activity, we have convened the BRAIN Initiative Working Group 2.0. That's co-chaired by Catherine Dulac and John Mansell. They're having a series of kind of going on a listening tour to talk to researchers about different areas covered by the BRAIN Initiative. They've had 2 of the meetings, one on the state of human neuroscience, and one about emerging opportunities. The upcoming meeting is next week, From Experiments to Theory and Back. This will be pretty computational. And these meetings are videocast, webcast, and the videocast is archived on the BRAIN Initiative website, so if you want to know what happened at the prior ones, you can go and watch it. It's available to all of us. And at the same time, there's also an RFI soliciting feedback from the research community as well as the general public. This is the notice. And you can, again, put that in your Google search bar and go and if you have strong feelings about what the BRAIN Initiative should or should not focus on during 2.0. The other thing in terms of data release, the BRAIN Initiative Cell Census Network, BICCN, I can remember that acronym, just released its first data set. This was focused on mouse motor cortex, molecular profiling for over 1.3 million individual cells from the mouse brain. The data sets that are available include single nuclear and single cell RNA-seq, single nuclear attack-seq, methyl-seq, and we have also completed molecular profiling. When I say we, I mean not me. Molecular profiling on 800,000 human cells. Some are post-mortem and some are actually from surgically resected tissue from epilepsy patients. And that data is working its way through the data release approval pipeline. It has a couple of extra steps to go to release human data to the research community. Staying outside, I want to talk for a couple of minutes about the Common Fund. The NIH Common Fund is under the Office of the NIH Director, Francis Collins, and the goal of the Common Fund is to fund projects that cross all of the institutes. They're supposed to be projects that lift off boats. The sort of project that inspired the existence of the Common Fund was the Human Genome Project, which obviously has had ramifications throughout biomedical research. And so there are a number of very specific projects. Dr. Lieberman mentioned the GTEx project that I was involved with. But I want to talk today about the high-risk, high-reward programs, which I'm sure many of you know a piece of some of these. This is a set of 4 different RFAs that are released annually. And I know you've heard of some of them. The Pioneer Awards support individual scientists, so they are very PI-focused, proposing bold approaches to major challenges in biomedical research. So these are cover the whole gamut of what NIH supports. They're open to all career stages. They do require 51% of your research effort, but the award is up to 700,000 directs per year for up to 5 years. The Transformative Research Awards are the one out of this portfolio that is not PI focused, but project focused. So multiple PI projects often do very well in this. These are for unconventional research projects with the potential to overturn fundamental paradigms, open to, again, all career stages, and the other differentiating thing about these is that there is no limit on the budget. So they are often very large projects, and therefore no prior approval required, no over 500K letter. And no preliminary data required. The New Innovator Award is our ESI award, Early Stage Investigator. It supports early career investigators proposing innovative and high impact research. An early stage investigator at the NIH is defined by NIH as being within 10 years of doctoral degree or medical residency completion and having had no R01 or equivalent grant. And in the RFA, it lists what equivalent actually means. Again, no preliminary data required. 25% of your research effort focused on this project, and the awards are up to 1.5 million for up to 5 years. So, yes?
Does the residency include fellowship or no?
No. And the Early Independence Award is the newest of these projects. It's programs that's still considered experimental. This is a program that allows early career scientists to skip the postdoc and enter independent research positions straight out of your PhD or your residency. Within the application should be in within 15 months of your terminal degree. And you need to be in a non-independent position at the time of the application. It does require the support and commitment from the institution, but not preliminary data. And we want 80% of your effort for the first 2 years of the project. And the next receipt date is tomorrow. So anybody who's watching on the video cast, I'm sure it's because you were putting in your application right now. Good luck. So just some highlights of projects that we funded last year. I should say these are annual receipt dates. They're mostly in September. The others have just passed. So if you're thinking about this as a possibility, now's a great time to start working up your project and thinking about it for next year. And meanwhile, if you're called to be a reviewer on one of these, thank you so much for your service. That would be great. Most of these, the review is they're conducted by CSR, and the review committees also span the breadth of biomedical research. And we usually on this main committee have maybe one neuroscientist serving. I've once seen a psychiatrist. And so anybody who volunteers to serve, I really appreciate your service. So some of the projects that we funded in the general area of neuroscience last year, Early Independence awardee Hilary Finucane is a mathematician at the Broad. She's using large genomic data sets to understand how GWAS signals translate into cellular functions in psychiatric disorders, so moving from gene changes to cellular phenotypes at least. Transformative research awardees Long Kai, Carlos Lois, and Michael Elowitz are creating methods to record cell lineage and gene expression in the individual cell. The acronym is MEMOIR, and I don't remember what that stands for. New innovator Priya Rajasethupathi, who's right down the road here at Rockefeller, is using outbred mice to link genetic loci to circuit changes underlying cognitive dysfunction. And then Amit Etkin is a pioneer awardee who's studying the neural basis of emotional disorders and their treatment. While I'm talking about innovative and creative and paradigm shifting initiatives, I wanted to say a minute about the NIMH BRAINS program, which is run by my colleague Kathy Anderson in the Translational Division. She is quick to say that she came up with the acronym first before the BRAIN Initiative. But this is a program for, again, early stage investigators to support innovative, creative, and ambitious research proposals that are really focused on mental health questions or disorders. The goal is explicitly to support promising new investigators and also to reduce the average age that promising early stage investigators receive their first R01. The eligibility requirements, we take projects that address any of the gaps in our strategic plan. Again, an early stage investigator by definition with no prior R01 support and an exciting and impactful project. And this offers 5 years of support with up to 1.6 million directs per award. And the next receipt date is next June, so now is really the time to start thinking about your application and get in touch with us. Said that a few times. Training. So we fund pretty much training mechanisms all the way across the career stage, from graduate or medical students. The R36 is for people from diverse backgrounds who are finishing their dissertation, F32 postdoctoral fellowships, various flavors of Ks. We have in my division mostly K99s, but also some K01s and K08s. I wanted to say there are a number of different ways that you can get support from the NIMH. You can get your support directly through one of these mechanisms as an individual award. You can get support from your institution through a T32 project, or you can get support through an administrative supplement awarded through on a grant to your mentor, either if you are an individual coming from an underrepresented background or re-entry candidate, somebody who's taken some time off from research and is trying to get back into the field.
Sue, can I ask a question? You know, at various points in time, depending on, I guess, funding exigencies and director preferences, you know, some mechanisms are more in favor or less in favor. And there was a time when the NIMH was concerned that they had too much of their portfolio in training mechanisms, whether it was Ks or, you know, T32s. Can you comment on what the sort of view of training awards is now? Is there a desire to keep them flat, reduce the amount, increase it, or what?
At this point, we are not looking to change the proportion of the budget that's going into training. We've kept it relatively flat. We don't have a strong push to increase it. We did, when Tom Insel was the director, have a period of where the level was reduced. But we're, at this point, I think, pretty comfortable and pretty stable. Yeah. So if you want to look at the information a different way, we have programs in different flavors. We have projects for PhDs, for MD PhDs, for holders of any kind of health professional degree, for people from underrepresented backgrounds, and projects for all kinds of applicants, and then, of course, the institutional T32s and short courses for training. And I realized I forgot to bring my own business cards, but if you'd like to talk to somebody about training, I have their business cards with me. So apparently that's what my subconscious thought was important to bring today. So the last thing I'm going to talk about is the budget. And I'm sorry, because I realized I am running over. We have had a relatively stable budget and success rate for the last 5 years. We're estimating that we'll be at a 21% success rate when fiscal year 18 finishes on Sunday. People at home are frantically pushing the last handful of grants out the door, so I don't have absolutely final data on that. I want to talk about the appropriation. This shows our appropriation in blue in actual dollars and in red in constant dollars based on 2000. We have been dropping for a number of years in constant dollars, as you know. And 1 year we actually had a budget decrease, which was a first for the NIH. But in the past few years, we've been competing very well in the national budget priorities, and even in terms of constant dollars, we are getting back almost to where we were at the end of the period of the budget doubling in 2003, 2004. Yeah.
Number of applications
it's fairly steady. Yeah. Yep. So what about for this year? This is not good news, but it's a good rumor. On September 13th, the House Appropriations Committee filed a two-bill appropriations minibus. The minibus is a take, when they do the whole budget at once, it's called the omnibus, but they do them usually in chunks. The Department of Health and Human Services that we're part of is grouped with labor, so our bill is called Labor H, affectionately. So it's also been approved by a joint House and Senate conference committee. The bill provides an increase of $2 billion for the NIH, which is something else, above the fiscal year 2018. For NIMH, the bill provides an increase of $98 million above the fiscal year 18 enacted level. And you read the same news reports I do. I don't have any inside information, but they suggest that there's a good chance that this bill will be passed by both chambers prior to the start of the fiscal year. This will be the first time since I've been at NIH, 22 years, that we've had a bill on budget on October 1st, if they do it. So fingers crossed, that would be really amazing. So what are we going to do with that money? Well, I just spent a little time telling you about where our interests were and what we do. This is what we did in fiscal year 2017 in terms of RO1s and RO1 equivalents. This graph is the applications that are awarded in blue by percentile, and in red, the applications that we didn't pay. And you'll notice that we state that our funding strategy is to pay 3/4 of the applications that score better than the 20th percentile. And we do that based on program priorities. We don't have a strict pay line. This doesn't equate to a 15% pay line, although you might think mathematically it would. So we reach for a handful of applications that are beyond the 20th percentile. We also skip in and out, a few below 10, a few more between 10 and 20. And those decisions are made through a series of consultations with program officers based on peer review, based on interactions within our division, with our division director, ultimately with Josh Gordon, and in consultation with our council. We are making priority selections based on research areas, gaps, opportunities, areas of the portfolio that we think are very well filled out. If your program officer has said, we have a lot in that area, they're giving you information, and that's important to listen to. Please get in touch with us. I keep saying that, right? This is a really bad day, both for us as well as for the applicant, and I don't like to skip applications that have done really well in peer review.
Do you know what the one that was 38th percentile?
Off the top of my head, no, I don't. It may not have, it was probably not in my division.
And obviously, you know, the more money, the better, but is there like a target percentile that the Institute feels is the most comfortable to be at in terms of percent being funded?
Better than we are now. There's a lot of good science that we're having to leave on the table.
So I think the maximum, at least in my recollection, was when it got up to 30%.
And when we were in the middle of the budget doubling, I was not in the extramural program, and so I wasn't paying as close attention, but yeah, they were funding pretty deeply and had a lot of flexibility. It was great. Yeah. We're doing okay now. A $2 billion increase for the past 2-3 years has been a big help. We are making decisions in alignment with the NIH Next Generation Researchers Initiative. We are prioritizing funding for early stage investigators and also using the little known but becoming more widely known R56 high priority short-term project to provide short-term support for some early stage investigators, but more at-risk investigators. Those are PIs who we understand are at risk of losing all their NIH funding within the fiscal year. We want to keep the workforce healthy and vibrant, and so we're trying to keep people going while they're resubmitting. As the end of the fiscal year 2018 approaches on Sunday, NIMH anticipates awarding 80 unique early stage investigators and more than 340 at-risk investigators for this fiscal year. I keep saying call me, call us. How do we find us? If you go on our webpage, this is the main page, there's a button at the top that says Research Priorities. If you click through, it'll list all the divisions. I'm under the Division of Neuroscience and Basic Behavioral Science. If you think your project is more translational, please get in touch with my colleagues in the translational division. If you don't really know, get in touch with me, get in touch with them. We will find the right person for you to talk to. And the last thing I want to say is that all of this information that I am giving you in not very compact form is publicly available. It comes from a lot of different sources. You know about the NIH Guide subscription for updates on new funding opportunities. That's across NIH. The NIMH Director's Messages, that's Josh's blog as we know it. It's about monthly, and he talks about things that we want to emphasize, things that might be changing. The council meeting open session, it's available on videocast. A lot of these slides came from Josh's update to the council meeting last week. And it's publicly available and archived. We publish a newsletter 3 times a year talking about what we funded and where we're going. And we're on social media, on Facebook. Josh and Jane Pearson in the Services Division just did a Facebook Live for half an hour last week about suicide and new research on suicide. Josh tweets, the Institute tweets, we have a YouTube channel. We're reaching out in all the usual ways. So thank you for your time, and thank you for your support of the mission of the NIMH.
Susan, thank you very much for that overview of the Institute's activities and funding opportunities. You know, as I was watching it, I was just curious as to know how, how many people are actually fully aware of the different opportunities. And I guess in that context, what's the best way for, institutions should be feeding information to faculty and trainees about funding opportunities. But do you have any suggestions or hints as to what you think are the most efficient ways to keep abreast of all the different opportunities? Other than what you just showed us on the website.
So there's the NIH Guide. We puts out, I think it's weekly, it's an email list, and it's literally the list of all the new funding announcements. There are program announcements, there are the RFAs, what's going on. The NIH, our open session to council, we do concept clearances. So if you want to know what's coming, usually when we present a concept to council, that's because we have an idea that may turn into an RFA, it may turn into a program announcement of one flavor or another. But it's an area that we're interested in, and so that is a great insight into what's coming. If you go on our council webpage, previous concept clearances are there and posted publicly, and you can take a look, and that will give you a great hint about what's coming along.
I don't want to sound ungrateful, but and I'm delighted with the fact that the Congress is doing the right thing. But if you look at the total federal budget, it's $4.3 trillion, and over close to 30% of it is in goes to HHS. The vast majority of it goes to CMS for funding Medicaid and Medicare. And that's one of the fastest. And so everybody thinks that the defense budget's the biggest thing. The defense is $500 billion. The HHS budget is $1.2 trillion. And most of it, like 0.9 or 1 trillion is for CMS.
Yep.
The NIH budget, as you just showed, is about 0.9% of the federal budget. The NIMH budget is like 0.04% of the federal budget.
Yep.
Goro.
Yep. And we're grateful for every penny.
Hi, thanks for talking the update on various NIMH priorities. So I'm an ESI, and I'm wondering about where you guys draw the line between the basic neuroscience and the translational neuroscience. So my background is in neuroimaging. I do human neuroimaging studies, trying to translate kind of basic neuroscience concepts about like visual scanning and ocular motor things to disease populations. And the thing that I've run into is that some people view me as a basic neuroscientist, or some people view me as a translational neuroscientist, and with that are different kind of expectations about output and just kind of general timeline of career development. So I was wondering if you can elaborate a little bit on that.
So my goal in our division is to not draw a line, because I don't think there should be a line, and because lines turn into walls. And what I hope is that what we have is a pretty fluid boundary. And with that, the expectations that go along with the actual research project and the question being asked. That's idealistic, I understand that, but that is our goal. And really, it would depend as an ESI. If you came and said, does my project go in your division or the translational division, I'd say, show me your aims. And I'll have you talk to the program officer who has the portfolio that it would be most comfortably housed in. I don't think we have different expectations about publications based on whether you're in my division or the translational division. I think we have different expectations based on what kind of research you're doing. If you're doing brain imaging, that's a very different kind of thing than if you're doing molecular biology. And we understand that in my division just as well as in the translational division. But if there's something specific that I can help you with, talk afterward, I'm happy to.
Having a bit of an identity crisis. You kind of had a period where you were focusing on animal models and model systems for brain research and medical. What's your opinion of iPSCs and organoids?
So we have a large and healthy portfolio using iPSCs and organoids. We have a guy, a program officer in my division, is David Panchison, who worked in that area as a researcher at the Children's National Medical Center, and then we were fortunate to be able to recruit him to manage a portfolio. There are definitely exciting things that had never occurred to me that are coming along in that area. There are a lot of technical challenges in that area, and David is really on top of all of them. I said earlier to somebody that I pay him to know that for me. And so it's not an area to just jump into because you're good at cell culture, because iPSCs in a dish have unique and unpredictable behaviors. But we see a lot of promise in that area for people who are willing to work through those challenges.
I have another I'll get to you, David, 1 second. Boundary question. So, you did a lot of work in developmental neuroscience, and, autism, Fragile X, Down syndrome, Rett-Williams, all these neurodevelopmental disorders are presumably, tractable using these various, scientific disciplines now. Is that NICHD? Is that NIMH? Where does that fall? And does that have the emphasis that it warrants?
So we have shared interest in a number of early developmental disorders. We are the lead institute for autism and the autism spectrum disorders. NICHD is the lead for Down syndrome. And then again, it really, depends on the question that's being asked. If the question in the research has to do with psychiatric complications of developmental disorders, that often will come to us. But the intellectual disability, primarily, issues will go to NICHD.
David?
So in many previous government research areas, like, for example, space, there's a huge movement toward non-governmental private enterprise to do all kinds of things that previously that NASA and JPL did. So just out of curiosity, is there, in the landscape, do you have any sense of how much is being done in comparable quests and missions outside of NIMH?
A lot. There's a lot of private money moving into biomedical research and into brain research in particular. And you've seen it here in terms of investment in the university from organizations like Chan Zuckerberg, from the Allen Brain Institute, and we've been, to the extent possible, collaborating with these organizations. They have definitely a role to fill. A lot of private investment is very focused on the interests of the investors, and probably as it should be. The traditional investment from pharmaceutical companies, for example, is really focused on small molecule treatment development and getting drugs out. The focus from philanthropic people has to do with their interests of their organizations. The role for the NIH is to make sure that we cover as broadly as possible and try not to take on bespoke projects, but cover the landscape.
Is the boundary fluid so that you go out to acquire these things, or, I mean, do you survey and acquire these things, and if they're worthy, do you, as it were, gobble them up and
No.
And you don't do any prospecting of what's going on out there?
We pay attention to what's going on, absolutely. And a lot of projects that wind up coming to us as R01 applications, the pilot data was gathered, for example, through private foundation money. But there are also private foundations who are doing very large initiatives that are kind of in parallel with us, and we encourage people to, if they're generating a data resource, please use that data resource and come in to us with a research question.
Thank you, very good answer.
Mm-hmm.
I'm just going to take the opportunity, since Jeff had to step out and he asked me to moderate, I'm going to take the opportunity to ask you a question about the role of animal models and the use of rodents, for example, in the NIMH portfolio. I had gotten the message during sort of towards the end of Tom's tenure that there had been a shift in the thinking of a program in terms of the best way to use mouse models, for example. Mm-hmm. Could you expand on that? Because there really was this, I think, just to set it up, I mean, the notion was that animal models, rodent models of human psychiatric disorders was no longer really thought to be an appropriate use of the model. They really wanted to focus the mouse studies on basic circuit-based questions and, fundamentally physiologic questions.
You've given me the answer, right?
Okay. So is that still the case? I guess so. And maybe add a little bit more about why program became soured on the notion of animal models as being useful for at least, obviously not in every instance, but in some instances.
Right. So I think I would reframe it just a little bit and say we reduced our interest in animal models of disease. That we felt like psychiatric disorders are in many cases uniquely human, and that the models that were being presented to us as a model of schizophrenia or autism or whatever, while it had some face validity, it was not teaching us about underlying circuitry necessarily. It was not teaching us anything useful about treatment development, and that we really needed to take a step back and saying, okay, what can these mice teach us, and what questions should we be asking of them that will tell us something about human disorders. So we really wanted to focus on as you said, the underlying circuitry, molecular biology, and questions similar to what I presented in some of those new initiatives that we're talking about, where the physiology questions model basic behaviors or physiology that we see in humans.
Thank you. Thank you. Jeff, you had one, and then Pete.
Hi, thanks for a great talk. Just a quick question. You spoke a little bit about the updating of RDoC. I'm curious how NIMH is thinking about funding translational and clinical research from an RDoC versus a DSM perspective these days. It's sort of a broad question.
That's a really broad question, and I'm in the basic science division. So I can give you a really broad answer and direct you to my colleagues. We are very much focusing on the matrix, on using dimensional characterization and following up on research questions that take that dimensionality into account. And if you have specific questions, I can help you figure out who to talk to among my colleagues.
Okay.
Hi. I had asked you earlier on about big data, and big data has been in the news recently with a couple of scandals about what people are doing to profit personally from big data. For the aggregate data in NIH, NIMH, from all of the studies that have been done, the question is, who owns that data?
Who owns the data? Well, the glib answer is that if NIH funded it, then NIH has a stake in it. But where those data are, realistically, and especially historical, is, as you know, a huge puzzle.
But does that mean that the investigator owns some of the data, or does the government own it because they financed it? And I'm sure this data resides somewhere on some servers or in the cloud somewhere, and there's lots of people that have a vested interest in mining that data for whatever purpose. So I'm just wondering who owns it.
So I think the question is, which particular data are you asking about?
Well, say a grant.
So
Someone gets a grant, they collect that data, the data resides somewhere, the investigators publish about it, but who owns that data? And then when you aggregate it, who owns the aggregate?
Right. So I mentioned earlier, genomics is probably the farthest advanced in terms of data sharing. In some ways, it's some of the easiest data to share, and in other ways, it's hard because it's all human data. And people are leery about sharing their genome. It's the ultimate fingerprint. We have a genomic data sharing policy that's NIH-wide. If you're taking a genome-wide approach, which is what we're doing for most of our studies, we ask you to share your data. The data is, by and large, our genomic data is in the database of genotypes and phenotypes, dbGaP, and accessible to qualified researchers through that. Ownership is, sounds like a legal question that I'm not sure I'm the person to dig into.
I can comment on this question. So Steve, for large studies particularly, a requirement of the conduct of a study is that a public access database will be created at the end of the study. You know, it's done for all the genetic studies now. That's what the Pat Sullivan's thing, what's it called?
The PGC, the Psychiatric GWAS Consortium.
Right, you know, so you can access, you know, tens of thousands of subjects' data in that context. In the KD study, you know, our deliverable was a public access database. For the smaller studies, it's really probably not useful to, you know, make them publicly available because what it would be, but journals are now going towards a policy. It hasn't happened yet, but they're going towards a policy that to submit to the New England Journal or to Lancet or to JAMA, the authors have to agree to make their data publicly available. And that's creating a lot of questions of logistically of how to actually implement that. And so the ownership has been skirted. So, you know, you're right, it is a legal question because if there's any intellectual property that comes out of the, you know, computational analyses, well, I'm not sure how that works out, but there's certainly a trend for the NIH journals, maybe other institutions as well, to try and provide, you know, publicly accessible databases.
All right, any other questions?
I'll ask one more if I could.
Yes, Dr. Lieberman.
So, you know, you talked about it at the beginning, you know, the process of developing assays that could identify, you know, are computational, but also assays that would be valid for identifying targets for therapeutic discovery and development. And that's been really an uphill challenge for the field, and the NIMH and pharma has certainly voted with its feet in that regard. So what's the, and I know you and Linda were actively involved in the CDDGs and, you know, the efforts to try and, with NCATs, what's the current attitude? Is it pessimism? Is it optimism? Is it, you know, we're going to, you know, trudge on, or we're not going to do anything? Because, I mean, and with Tom had restricted, you know, any therapeutics grants to really just a minimum, a single mechanism, you know, that was in a very early stage. But if you could just share with us the current opinion or current view.
So we have an active portfolio in assay development, treatment development. Lois Winsky has been pushing on this for longer than I've known her. Like I said, I talked about 2 new initiatives to try to push for more physiological focused assays that are more parametrically defined. And that's part of, I think it's a really hard problem. As you said, pharma has voted with their feet. This is where NIMH needs to step in and see what we can do. We are trying to come up with what we think are the logical next steps or pre-steps. If we need to work backward in the pipeline to develop more animal assays that are ultimately translatable, that's what we're trying next. But Lois has really devoted her career to try to help solve this problem. And there are others also in the division who are working on the preclinical assay development.
But apart from the R6133, what mechanisms are there now?
There's got to be an easier way to do this. Does this work? No. So I talked about these the PAR that she's got out, which is it's a cooperative agreement. I think it's a UG1 UM1. It's some weird mechanism that I don't. Yeah, this is one, which is about validating assays between human and non-human systems. And the NCDDGs that are also out, that are available as options.
So it's all in very early stage development.
That's what's in my division. Right. So I can't speak to what the translational division is working up next. I'm sorry. Ask Josh, ask Holly.
Holly's very, she's also very heavily invested in brain stimulation.
Yep.
Is that him a crossover to your division?
We do sort of a fair bit of basic science in terms of brain stimulation and modulation in circuits in non-human primates and rodents. Yeah, we have our learning and memory portfolio. Bettina is really interested in this area, and we have an ongoing PA in that area. Yeah.
All right.